Protein is a structure that occurs in 4 parts. We have primary structure, secondary structure, tertiary structure, and quaternary structure. And we're going to go through all of those. And this Play-Doh that I made here, these various colors, are going to help me demonstrate each of these structures. The first one is primary structure. It's a sequence of amino acids, and the sequence is determined by DNA. As you can see, here we have a chain of amino acids, and each little ball represents a different amino acid. So, secondary structure is when the polypeptide is folded and coiled. This is due to hydrogen bonds between the backbone. There are 2 structures that we're going to go over, the alpha helix and the beta pleated sheet. The type of amino acid within the polypeptide determines the specific structure. So in the alpha helix, we have amino acids hydrogen bonded to every 4th amino acid. So these amino acids are the pink, and there's hydrogen bonds between every 4th. And this pattern kind of pulls the polypeptide into a helical structure that's coiled. And since my Play-Doh is so bad, I have it on a straw. But you can see it kind of looks like a curled ribbon. And this allows the R groups to stick out freely. So then we also have the beta pleated sheet. And there's segments of the polypeptide that line up to form a sheet. And there's hydrogen bonds between the parallel segments. So if this was another polypeptide, it would kind of hydrogen bond right there. And you can see it'd be kind of like a sheet. And there's some amino acids that normally form these beta pleated sheets, like tryptophan, tyrosine, and phenylalanine. And there's some structures that actually don't take on either of these shapes. For example, proline is found in unstructured regions of the protein. All right, so the tertiary structure is the overall 3-dimensional structure of the protein due to interactions between R groups and bonding. So you can see here we have these amino acids, and this yellow is the side chains, and the bonds between them kind of form this overall 3D protein looking thing. And then we also have 2 special properties that align with this structure. So the first is disulfide bridges, where there's covalent linkage between the sulfur side chains of cysteine. So if these pink balls are cysteine amino acids, and these are the side chains, the sulfur side chains, they kind of interact and bond there, which forms really strong bonds within the protein. And then we also have hydrophobic interaction, which is where the hydrophobic parts of the polypeptide cluster on the inside, so this pink right here, and it kind of leaves the hydrophilic parts, the blue, on the outside, which allows for the protein to interact with water. And then finally we have our quaternary structure, which is when there's multiple polypeptide chains that form and come together. So not all proteins take on this structure, but when they do, we have lots of our different structures like our alpha helix and maybe our beta pleated sheet, and maybe like another alpha helix, and they kind of come together and they form a big protein, which is kind of hard to show with this Play-Doh structure here. But if you want to think about it in terms of like the colors and everything we used, with our alpha helix we had pink, and the beta pleated sheet we had blue, and it's kind of like if you had like a big jumbled mess of those colors, and it just kind of folded up and formed this whole entire protein looking thing. And this really shows what our protein is. As the final result of all of these structures. So next time you have to think about the structure of proteins, maybe think about it in terms of the different structures we showed, and our amino acids and the chains, and maybe some of the properties that we talked about with our Play-Doh.